Great, so we're going live. Thank you everyone for joining us today. This seminar is part of our open source neuro series, which in turn is hosted in the Worldwide Science Initiative. The series is all about open tools and methods in neuroscience that are on our website, open-neuroscience.com. You can upload your own projects to the website and sign up to give a talk by completing short forms. We welcome your contributions, and if you can share our content and follow us on Twitter. You'll find all relevant links to do this in the chat. Today we have the pleasure of hosting Dr. James Knight. Dr. Knight originally studied electronic engineering at the University of Warwick before leaving academia to work in the computer games industry. After 5 years of that, he got bored, those were his words, and started an MPhil in computer science at Cambridge, where he first encountered spiking neural networks. Based on this newfound interest, he went on to study for a PhD at Manchester, where he worked on simulating plasticity using the Spinnaker neuromorphic supercomputer. After this, he moved to Sussex to work as a postdoc on the Brains on Board project, during which time he became the lead developer on the GeNN simulator and also made some cool robots. He is now an ESBRC Research Software Engineering Fellow trying to push biologically inspired machine learning with spiking neural nets further into the mainstream. We'll now listen to James talk about GeNN, and you are welcome to ask questions in the YouTube live chat or directly on Zoom. Thank you, James, for being with us today, and please go ahead.
Thank you so much, Cecilia. It's a great intro. So as you said, I am James Knight. I'm here to talk to you about GeNN, which is our GPU-enhanced neural network software. So, first problem. So, first of all, I'm going to give a little bit of background about spiking neural networks, because I realize they're not that common. Then I'm going to talk about ways people have and do accelerate simulations of these models, because they can be quite computationally intensive. Then I'm going to talk very briefly about GeNN, which is our tool which aims to do this. And finally, I'm going to talk about some recent projects both myself, other people in our lab, and some sort of outside users have done with GeNN recently, which I think is really exciting. So first of all, spiking neural networks, what are they? And also why? Why spiking neural networks? So from a neuroscience point of view, lots of real neurons communicate using spikes. Sometimes you want to model that, you need a tool to do so. And spiking neural networks provide a framework where you can model these systems. Also, from an engineering point of view, real neurons, especially in like the cortex, they only spike very infrequently, so kind of they emit a few spikes every second. With the right type of hardware and software, this can save computation and energy, because you're only communicating every few seconds rather than continuously, like in a standard neural network. Additionally, the exact timing of spikes contains a lot of additional information beyond their rate. I mean, some systems, like the kind of barn owl sound localization, relies entirely on this kind of exact timing of spikes, but it's a common sort of computational principle throughout the brain. And in the world of machine learning, this is really exciting, because current machine learning networks are operating on this abstraction where you have a kind of real value number which represents the sort of abstract rate of a population of neurons. Whereas if you have an exact spike timing, that this exact timing of the spikes within that can contain a lot of additional information and hopefully enable kind of new exciting machine learning applications. So what does a spiking neural network actually look like in the sort of lowest level? So you have some neurons. Unlike the neurons in a machine learning network, they tend to have some state. So this is what's called a Zakhaevich neuron, and this has got two state variables with some dynamics described by some differential equations. So they're coupled, and there's some discontinuities as well. So with the V state variable, which is a membrane voltage, roughly speaking, if it reaches some threshold, then there's a sort of artificial jump is induced in both state variables, and a spike is emitted. So this results in this kind of voltage trace you can see in the top right, which roughly speaking resembles a regular spiking neuron you might find in cortex. So these, if you've done any kind of programming work, these are very simple to implement. Like really, you just have to solve this equation. They're all independent, so there's no problem in parallelizing them or anything else. But these neurons are connected together with synapses, and these tend to be sparse. So all your neurons are not connected to all your neurons. So you have some kind of sparse connectivity, where so this neuron is connected through this synapse back to this neuron, and back through this synapse back to this neuron. So this is kind of a sparse updating problem. This is much trickier. So this is where a lot of the challenges of simulating these models come from. And how do we use these to model brains? Well, we kind of tend to approximate sort of anatomical regions into groups of neurons with similar properties. So, for example, on the right-hand side of the screen, this is a picture of a model of the sort of generic insect mushroom body for olfactory learning. So this is the antennal lobe, which is connected to the antenna, where the olfactory information comes from. There's a lateral horn where there's inhibitory interneurons, and there's a main body of GeNN cells here, and some output neurons whose acronym I can't remember. But another important property is that in biology, neurons have many connections. So each neuron has up to sort of, in the cortex, 10,000 connections per neuron. So the synapses vastly dwarf the number of neurons. And this is why simulating synapses is such a tricky challenge. Back to our model, these connections tend to be, in this abstraction, the connections tend to be concentrated between the groups. So you've got some connections within a group. So this output neurons are self-connected. And then most of the connections are concentrated sort of between the groups aside from that. So there's sort of structured connectivity of some sort. And these, when you're modeling these systems, they tend to be modeled using sort of standard patterns. So you have a kind of fixed probability where you've got 50% here. So this means if you think about each possible pair of antennal lobe and mushroom body neurons, there's a 50% chance of there being a synapse there. So these are the kind of standard building blocks that you might model brain systems like neural networks with. So how do we accelerate these simulations? So because there's no kind of obvious fit for this problem, there's no like special piece of hardware that's being generally available. To do this, the typical solution is to use like big clusters and supercomputers. So these are just like lots of powerful CPUs, but they take up a lot of space. So this is a K computer in Japan. As you can see, it's pretty vast. And if you think about a neuron being simulated here and a neuron being simulated over here, there's going to be a lot of wire going between them. And so that there is inherently some kind of latency in going through all the switches along this wire to get communication across this. And this essentially limits the speed of simulating neurons in these systems. You spread out the problem enough that you've got enough memory and it runs fast enough, but then you're always constrained by the communication between the nodes. And also these systems are very power hungry. These type of systems require megawatts of power to run the whole thing. I think this is a K computer in Japan, by the way. So the far other end of the spectrum, we have this thing called neuromorphic hardware. So these are chips that are kind of built specifically to simulate the brain. And as you can see, they look remarkably like my diagram in the first slide. You have little circuits representing each synapse, and you have more little circuits representing each neuron. And in some of these systems, these are like literally analog circuits that mimic the behavior of neurons. Back in the 60s, they discovered that the sort of voltage current relationship of neurons is quite similar to the voltage current relationship of transistors. So you can make a little neuron circuit with a very small number of transistors that behaves very similarly. And this is amazing. These systems use tiny amounts of power to simulate neurons. And it's a very cool field. But nonetheless, you need to build a physical circuit for each synapse and each neuron, which means that it's not totally clear how these chips really scale beyond relatively small systems. And it's difficult to design. There's a very niche sort of electronics. Building these analog neuron circuits is super specialized, and it's very inflexible. Once you've built your system with however many neurons and however many synapses, if some irritating neuroscientist comes along and is like, hey, I want a different state variable in this neuron, then you've got to go back to, if not the drawing board, shortly afterwards, redesign your circuits, rebuild your chips, and all of this takes a long time. So in our group, we've been looking at using GPUs instead. They've got lots of advantages. They're relatively cheap and easy to obtain. And if you're forced by a university photographer, you can pose with them, like me and Thomas are doing here. And also they're available in a lot of form factors. So you can get little tiny embedded GPUs, kind of this big, that you can put in a robot, and you can get these kind of big standard GPU-looking things that we're holding that you can stick in the server. And they have lots of parallelism. So for example, an A100, it's kind of a modern data center GPU, has got almost 7,000 cores. This means that it could be simulating 7,000 neurons at once. And this is obviously really good because that means you can simulate them faster. And from a kind of pragmatic point of view, as people that are mostly software people and not people that are willing to spend their lives making chips, someone else is responsible for taking the latest and kind of chip technology and turning that into something usable. And as you can see on the left, GPU manufacturers have done a remarkably good job over the sort of last decades of scaling this technology. So I honestly can't remember which color is which, but one of these lines represents performance, so like how much maths you can do per second, and the other is memory bandwidth, it's like how much data you can actually get onto your GPU, because it's not much use being able to do lots of maths if you can't actually get any data to calculate things with. And these have both risen in together, which is great, and it's continuing to grow. Every generation of GPU gets better. So our software can stand still and it still looks better year on year without any effort from us, which is the dream situation. So how do you map spiking neural networks to GPUs? GPUs look like all hardware have got kind of a regular structure where you launch like grids of which are all performing the same task, and within that you've got lots of little threads, and these are grouped together into blocks. And it's beyond the level of this talk, but the key thing is you've got to kind of round things up to fit into this regular structure. And as Thomas Novotny, my co-author and colleague, says, this is all of bean counting. This is like really not very interesting work. So this is the kind of thing that software should be taking care of for you. And this is where the origin story of GeNN came from. Back in 2009, Thomas got one of the first CUDA-powered GPUs. So CUDA is like the programming language used to program NVIDIA GPUs. So he implemented a model of factory learning network that we showed a picture of previously. So this was a kind of fixed size, Drosophila size, in fact, with about 2,500 GeNN cells in the middle. And after he learned CUDA, it took him about a month to implement a single model. And this was optimized for a single GPU. And especially back then, the kind of architectures were changing very quickly as they tried to figure out how to build better GPUs quickly. So by the time the next one came along, a lot of this model has been reprogrammed. And also, if you wanted to investigate how the learning situation improves, if you have a B-sized mushroom body with sort of 10 times more GeNN cells or whatever, you'd also have to reprogram this model and do a lot of the bean counting again. So this sort of bespoke CUDA programming is clearly not the way forward to make a useful tool for computational neuroscience. So this is where GeNN came along. So it was originally just a framework to solve this very problem. And on that basis, it's got a couple of kind of maxims, which we've tried to stick with to this day. So the user's got maximal control. They can use whatever equations, non-biologically plausible updates they want within their neurons. You just have to type the code in. And what GeNN does is generate the kernels to simulate these on the GPU and helper functions to transfer the data backwards and forwards. It's up to the user when to actually call these things, so when to copy stuff, and what to do with the data they've got off. So there's no kind of standard file format, so you must use or any of that. You can integrate it very tightly with your code, which can be great. And also the user can interfere, so you're not stuck with whatever models that we think you should be using. You can provide your own. And this is kind of more for developers really, but the code that GeNN generates, because it's a code generator, so you run GeNN and it builds you some CUDA code that simulates your model. The code that it generates is human readable after a fashion, so you can interfere with it. If you're thinking of adding something, you can sort of prototype it like that. There's obviously, that's great in a way, but people don't like directly driving everything from C++. So we've kind of expanded this ecosystem around GeNN sort of upwards and downwards to sort of cater to what people actually want. So on that basis, we can now generate C++ code, so you can test your model on the CPU if you don't happen to have a GPU with you. You can also generate OpenCL code, which runs on kind of Intel and AMD GPUs, although this is a bit experimental still. And you can generate CUDA code for NVIDIA GPUs. And on top of this, there's also kind of more friendly ways for users to use this system. So there's Brian2GeNN, which interfaces GeNN with Brian2, which is a very popular open source brain simulator, or spiking neural network simulator rather. And that's been a kind of ongoing project for some time. There's SpineML2GeNN, which is an even worse name than the other ones. But this interfaces it with a kind of graphical neural network tool called SpineCreator, which is developed at the University of Sheffield, which is also very cool. And finally, we've got PyGen, which is kind of a direct Python interface to GeNN. So we've tried to keep by following the same maxims. So it's still very, very flexible, but you can do everything from Python, which is frankly much nicer than using C++. Even we, the authors, kind of stopped using GeNN directly from C. It's all about PyGen these days. And then because it's in Python, you can interface with all these other cool open source tools. So you can interface with PyNN, which is a sort of human brain project standard for spiky neural network definition. And we've more recently been developing this thing called MLGen, which is intended to be kind of a tool for solving machine learning problems using spiky neural networks. And this lets you do all kinds of stuff, but currently its main use is that you can train models in TensorFlow, so kind of standard spiky neural networks, and you can convert them to spiky nets to see how that affects the performance and sort of prototype things for maybe putting on neuromorphic hardware in the future. So that's the kind of background to this. And the rest of the talk, I'm going to talk about projects that use GEM. I see there's some questions in the chat. Okay, cool. It's just Andre, that's fine. I'll stop micromanaging. So first of all, this is some work that I've been doing over the last few years. So we've been looking at these cortical microcircuit models. So this is a model of a cubic millimeter of sort of generic rodent cortex, and it's assembled from a whole kind of fusing a whole lot of data. So there's microscopy, which is used to find the density of synaptic connections between the regions of cortex, and so this is all expanded out with heuristics to give estimates of the connection densities between the different cell types. So this is a 4-layer model of the cortex, and it's sort of abstracted away. So on each layer, there's one excitatory and one inhibitory population. And then this was paired with sort of calcium imaging of awake animals. So they had firing rates for each layer. So the model was kind of fit to obtain the same firing rate. So essentially, you get something that, when you simulate it, sits in a resting state that resembles what you see in awake animals. And this is kind of, at this point, more of a benchmarking model than anything else. It's not super interesting, I would say, but it has been used as a kind of basis of comparison between different simulators, because it is a real model. It's not just sort of one population of neurons connected to themselves. It's sort of much more realistic, and it's quite big. So it's got 80,000 neurons and 300 million synapses. So as I say, there's a lot of synapses for each neuron, and all of these, because all this data is from biology, there's always some uncertainty. So the strength of each synapse and like the delay of each synapse is modeled with a Gaussian. So there's kind of a mean and a variance for each synaptic strength and delay. And back when we started this work in 2018, this was taking around 3 seconds to simulate 1 second of biology on a supercomputer. So we've been through a few iterations of this, but in short, this works really nicely on GEM. And on a modern GPU, it takes less than 1 second to simulate a second. So you can simulate it faster than reality, which is really, really exciting for kind of, if we added learning to this, kind of exploring how learning evolves, because if learning has to operate on biological timescales, then you have to wait a long time for things to happen. Along the way, the final kind of hurdle to getting to this performance, on this graph on the left, we're showing quite a lot of stuff, but mostly this is the performance of this model running on a desktop GPU, which is this GTX 1650. So this is the kind of thing you could buy for £100 and put in your terrible university computer, and it would probably work. This Jetson Xavier NX is a robot, sort of small embedded GPU for robots, and this Titan RTX is a sort of workstation GPU. So you can see that on all these systems, you've got some time spent simulating the synapses in orange, some in blue simulating the neurons, and then you've got this overhead. So this is sort of time that was being spent sort of copying spikes off the GPU every time step, that type of thing. And as we got faster, especially on systems without very good CPUs, so without very good processors like the Jetson, you ended up spending a lot of time copying data off the GPU onto your CPU for recording. Like this was on this, if you're doing, especially if you're doing in Python, it begins to dominate the whole cost of running the model. And this is what's called Amdahl's law. It's basically that you sort of optimize everything that's easily parallelizable away, and then you're left with this stuff that's not easy to get rid of, because you can't necessarily parallelize it. So we actually did manage to parallelize this. So we offloaded this recording to the GPU, so it's kind of keeping track of the data so it's keeping track of the recording data for the whole length of the simulation. At the end, you can copy the whole thing up, and this makes a huge difference. And this is how we ended up with a situation where we're getting faster than real-time performance. And because of this, Python and C++ doesn't really make much difference. You can still run the model really fast. And one note here is this is using about 10 times less energy than a supercomputer. Because energy is a product of power and time, the fact that you're using something that's using less power to start with gives you an advantage, but if it's faster as well, then you get kind of a even further advantage. So we then went on to look at this larger model. So this is a model of the macaque visual cortex. And each of the 32 areas that are modeled are represented by a kind of scaled version of that previous model. So the proportion of neurons in each layer is scaled, as this figure here shows. And all the populations are connected together based on these connectivity data sets. So as you can see, there's multiple sources that fuse together. So there's kind of this binary but fairly complete data from COCOMAC, and then there's this much more detailed, so it's got kind of connection strengths and densities, but much more patchy. So it's totally missing strips of connectivity here. And these are fused together using the algorithm these guys developed to give sort of connectivity between the regions. And this is combined with our knowledge of how cortex connects. This subfigure here shows. So this resulted in a massive model. So 4 million neurons and 24 billion synapses. And back when these guys were originally doing this work in 2018, this took 12 minutes to simulate a single second of model. But this, as I said before, this is mostly because I have to spread this out really thinly. These IBM systems only have 16 gigabytes of RAM on each like processor, which these days seems ridiculous, but they did. So it needs to be spread between over 1,000 nodes, and therefore there's a lot of wires and a lot of communication going on. And if you do this on a modern supercomputer, so this Eureka thing is probably about as fast as the CPU supercomputer gets. It's down to 30 seconds to simulate 1 second, which is obviously a significant saving. And this is also on many fewer nodes, but this is because they have so much more memory. So our question was, can we do this on a single GPU? This would be really cool. Lots of people would be able to play with this model for the first time without having access to a giant supercomputer. However, we did the kind of back of the envelope calculation. Each synapse requires somewhere around 8 bytes of memory to represent. So with 24 billion of them, you need about 180 gigabytes of memory. This is somewhat less GPUs now than it was then, but that at the time it seemed a very large number, something like 8 GPUs, which is obviously not quite as hard to get hold of as a supercomputer, but you're not going to be able to get 8 GPUs just to play with a model. No one's going to fund you for that. So we kind of went back to the drawing board a little, and if you've been really looking carefully at this talk, then there's been various bits in bold where I've talked about estimates of connectivity density, like Gaussian connection strengths, all this type of thing. So all of these, when you first run the model, traditionally all of these would be turned into actual weights for the synapses and like starting values for the neurons with very simple algorithms. So mostly you'd sample from a random number generator and do some algorithm to scale the result. So the idea here is, how about if you run these on the fly? So every time you get a spike, you recalculate the neurons that connect to it. So if it's 50% connected, you roll those virtual dice again and again to see which neurons it's connected to, and then you recalculate the connection strength for each one. And this seems like a ridiculous idea, but on a GPU, although the computation and the memory have grown, it still requires around 10 maths operations per byte of data read from memory for the usage to be truly balanced. So fully use the GPU, you need to read 1 byte from memory, then do 10 maths operations on it. So there's quite a lot of scope to get rid of some of the reading from memory and do some more maths. And that's essentially what we did here in this 2021 paper. And the results were quite good. So this is the kind of activity you see from this model. You're beginning to see something that resembles slightly less boring activity. You're beginning to see some kind of upstates propagating around the model. And the sort of overall statistics now match the original simulations that were done on the CPU simulator here. So these are 3 measures of spiking activity, and which the distributions essentially are a reasonable match. And the overall speed up we got to 7 minutes per second on a single GPU. So clearly a supercomputer is still faster than this, but it's faster than a previous generation supercomputer, and you can run this model on a single GPU, which is kind of the main goal. The fact it's quite fast is sort of cool. But so that's the end of my work. And I talk about stuff other people have done. This is going back to Thomas is very keen on olfactory receptor modeling. So this is a model of the bee olfactory system. And this is done in this paper where they were investigating how bees respond to this compound called geosmin. So it's a compound produced by bacteria and fungi. It's sort of got a rotting sort of smell, sort of musty smell, and it's strongly repellent to Drosophila, presumably because it means the fruit that they're trying to eat are rotting, so they're no good. It's attractive to yellow fever mosquitoes who want to lay eggs in it. Maybe it looks like a swamp, I don't know. It's unknown what it does on bees, and I think even after this paper, it's still not 100% clear what it does on bees. But this paper was quite cool. It combines behavioral studies on bees, electroantennography, which I can only just say, which is basically recording the voltages coming straight from the antenna into the antennal lobe, as well as a computational study of this system. So for this computational study, you have kind of three types of neuron. You have these receptor neurons in the antenna itself, and these project to the projection neurons, which is the first layer of the model that I sort of talked about in a few contexts previously. But these have got some very interesting inhibitory connectivity between them. So it connects to interneurons, and then the interneurons connect back to all the other. So each projection neuron is connected to the interneuron. The interneuron population then connects on to all the other projection neurons and all the other interneurons. So the first stage of this work was comparing the antennal lobe model. So this was done with sort of leaky integrated fire neurons, which are kind of very simple neurons with a spike frequency adaptation. This means that if you put the same input into the neuron over time, the response will reduce. This is something you see in a lot of neurons in the real brain. And these models are driven by this two-stage binding activation model, which is quite a specialized kind of olfactory receptor modeling thing. It's not something you'd find in a standard neural network simulator. So this is something we have to add to the model. And this is the type of thing that GeNN's really well suited for. Like you can add some custom logic to your model without a lot of difficulty. And this is comparing the electroantennography of the real antenna versus the spikes coming out of the receptor neurons modeled. And you can see that as the intensity of this geosmin compound geosmin compound increases, the response both in the model and the real thing increases, as you'd expect. So going deeper into the model, we're looking at calcium imaging of the glomeruli, which are the sort of groups of projection neurons that all connect to the same thing. And you can see that it's slightly hard to compare these figures because we're modeling 160 glomeruli, whereas in the calcium recording, it's only recording a subset of these. But the main thing you should be looking at here is that when the stimuli onset with these dotted lines, with a low concentration, so 10 to the -6 concentration of geosmin, you get sort of very selective activity in the glomeruli. But then, intriguingly, when you increase the concentration, this sort of spreads out and goes away. So on average, you've probably got a bit more activity across. You've got more glomeruli active, but there's no kind of strong pattern here. And again, these were modeled with neurons connected with conductance synapses. And this behavior is sort of repeated in the model quite well. And the logic is interesting. So as more glomeruli are recruited, so they're kind of less strongly. This is a fairly non-selective odor. So a lot of the projection neurons are recruited to this, and then the inhibition kicks in more and more because the inhibition is connected not just to the glomeruli, but also to the other inhibitory interneurons. So basically, the inhibition totally takes over and inhibits everything. So if you get too high a concentration, everything is inhibited, which is a really interesting effect. It's interesting to see what that would do, what that means behaviorally. But I'm afraid that this paper, I don't think answered that. So on to something entirely different. So this is a project from.
So let me just ask you a question. So you can go and inspect each of these neurons in the model itself as well, right? You could go and ask those questions.
Exactly. Yes, you could. Yeah. I think the question more was it wasn't clear what the bees were actually doing. The model kind of matched up to that point, but even in a bee, there's a lot of brain between there and actually taking the action. So the behavioral impact, what the behavioral context of this compound.
But the model itself is fully infectable, like you can go in.
Yes, indeed, that is the difference. Yeah, I mean, you can that amount of calcium imaging is about the limit you can get in a behaving insect, whereas a model is fully inspectable, which is why in silico neuroscience is cool. So this is a project with a postdoc from our lab, Garibaldi, and Laura Kleiner from University of Bern, and they were looking at this model presented in NeuroIPS in 2018, and basically they were showing that you could, the kind of cortical microarchitecture that you see, where you've got bottom up input and feedback input as well, how this can actually be used to for machine learning. So this is a model. This is inspired by this cortical architecture, and it consists of pyramidal neurons with 3 compartments. So you've kind of abstracted away the full complexity of a pyramidal neuron, but you have a basal compartment which receives a bottom-up synaptic input, an apical compartment that receives sort of lateral input from other pyramidal cells, as well as top-down input. And this computes an error signal across the neurons. So the kind of difference between the feedback and the feed-forward is the key signal here. And then the soma produces outputs. And these are interconnected with interneurons, which have just got a basal compartment in the soma. So this is showing that you can be very flexible with GeNN neuron models. You can start adding small numbers of compartments. Like, it's not a simulator to compete with neuron in sort of hundreds of compartments connected properly with a cable theory. But for kind of small numbers of compartments, you can do lots of stuff. Initially, they implement this model in NumPy. So this is the red line here. And you can see as you get more neurons, it gets very slow. But they then implement it in GeNN. This is before they turn into a spiking model. So this is still a rate-based model, which isn't really what GeNN's for, but still, you still got three orders of magnitude of speed up over that, which makes a big difference. Like 10 to the 5 seconds is a long time. 100 seconds is not very long. And even if you didn't have a GPU, GeNN still makes this faster than NumPy, but that's more just the advantage of C++ over Python, really. So what they were interested in doing with this, so this 2018 paper was showing that this architecture can approximate error-backpropagation. So this is the algorithm that's used to train a lot of deep learning models, but it requires these sort of exact feedback connections all the way through the model, which is not what you see in the brain. But the key thing that this 2018 paper showed was that this architecture can solve these credit assignment problems. So the credit assignment problem is that you know exactly what the synapse many layers deep in your model, how much error in the output that's responsible for. So they showed that this microcircuit-based architecture can solve these credit assignment problems in sort of standard supervised learning tasks. One of the tasks they use is this what's called a yin-yang model. So you've basically got 3 classes of output. You've got the eyes, you've got the yin and the yang, never mind which is which, and they're kind of encoded in the spike times. So this is like a very small, easy benchmark, but the key thing is you can't linearly separate it. Like if you draw a line through this model, there's no way of linearly separating it, which makes it better. It makes it a better test of kind of deeper models than MNIST, say, which you can separate linearly to a reasonably high degree of precision. So you haven't got much. It's very easy to get very high precision on MNIST. But they showed that this GeNN model, this can get 95% accuracy, which is pretty close to what people get with deep learning. So I think it's a very cool project. It's very exciting to see GeNN used in this way. Another project, this is Felix Schmidt from the University of Cologne. He's been looking at these clustered cortical models. So in the cortical models we talked about before, each kind of layer of the cortex is represented by a single population. So there's no kind of complexity in the connectivity there. And that kind of means that nothing really exciting is going to happen within your cortical layer because everything is connected to everything. There's no kind of local connectivity. And so I think a key step to actually getting these models to sort of represent stuff is to have some kind of clustering. So the excitatory and inhibitory connectivity is sort of denser locally, and the models split up. So you can sort of start seeing these wandering attractor states that you see in real cortical recordings. So this is divided into, in this case, he's got 20,000 excitatory neurons and 50,000 inhibitory neurons, and they're divided into 20 clusters. And he was interested in exploring the parameter space here. So this is the kind of parameter space you see. So you're changing the, these are the spike rasters. The red is the inhibitory, the black is the excitatory, and you're seeing the sort of different dynamics of these up states, so these sort of dense patches of spiking as you vary the sort of external stimuli into the inhibitory and the excitatory population. So you can see this is the kind of parameter space you're exploring. So somewhere around here, you're getting really nice wandering up states. Up here, where you've got very high excitatory input, you kind of get locked into a single state. This is quite a large, this ends up being quite a large parameter space, and this is only showing every 8th sample of each axis, I believe. So he was looking at these, he was actually looking at 40 by 40 grids at this point. So this is large, very subdivided models. And on this server, he was using a 24-core server using Nest, which is a CPU simulator. It took about 12 hours to explore this parameter space. Using GeNN, he got this down to about an hour. And there's some irritating bug that we still haven't solved that should have meant it was even faster, but still. So this is also really nice work. And he is giving a talk next week at a workshop called NICE, if anyone's interested in this stuff in general. I think it's very cheap to register online. It's a nice workshop. So that's the end of my talk. I just want to insert an advert here. So when we talked about the kind of libraries built around GeNN, some of them had this logo on. This is the logo of Google Summer of Code, which is a project, it's a program where Google pays for paid students kind of a relatively generous stipend to work remotely on open source projects like GeNN over the summer. And we've teamed up with INCF, who are an organization that promotes open source neuroscience for several years now. We've had students work on all kinds of projects. So if you know anyone, if you are a student who'd be interested in this or know anyone who works on this, this is exactly the right time to be applying because the deadline closes next month. And we'd love to hear from you. And if you've got any questions, please ask. Yeah, I hope that was interesting for you all.
Thank you so much for that wonderful talk. I think it was super interesting. And would you like to give us the Google Summer of Code if it's a direct link? If there's something you want to promote?
Yes, I realized I was practicing, and that'd be very useful. I just get can YouTube see see the chat?
No, but I brought it through, so that gets on there. It was amazing. I think that you showed, you went completely in depth of how powerful and completely customizable this is, and with all that interoperability, and also the in-depth breakdown of the costs of implementing each thing. So with all the projects that made it really, I think, easy for someone watching the talk later to understand and reach out to continue.
Fantastic. I'm so glad. Yeah, it's been quite a recent thing. We've actually got people doing interesting stuff with that outside of a lab, and it's really cool to see.
It's amazing. The variety.
Yeah, exactly. So this link is to it's not a very useful link, but it gives you a forum of all the INCF projects. Gives all the INCF projects this year, and there's several GeNN projects in there, as well as other cool open neuroscience related stuff.
Yeah, thank you so much. I hope they reach out to you. We can also write something up.
And in the past, it's been quite limited, what's the word? Criteria for who can apply, but it's now very open. It's kind of anyone that's interested in open source. So you don't need to be an undergraduate student with a particular institution. Like anyone who's interested in open source can get involved. And as such, there's some projects that don't require lots of programming knowledge to get involved in.
Okay, so I have a couple of questions if you're up for it, to get in a few things you mentioned, like this modeling of learning, like at longer time scales. Is that something that you're interested in, or that how far can GeNN go with modeling longer time scales?
Yeah, this is a good question, because I think biological learning clearly happens over very long time scales. So you need to be able to simulate a model really fast. We've done some stuff with sort of relatively small models, so kind of thousands of neurons, and we've had them running sort of hundreds of times faster than reality, which definitely goes in that direction. But yeah, there's not yet a good solution to that, because like if you start sort of cheating to make the learning happen faster, then it's not the same it doesn't match the dynamics of the neurons the same way. So it's definitely not a solved problem.
Yeah. Yeah, no, I was wondering like whether you could like interrupt the learning and then like simulate it starting later or something weird, but
yeah, I mean, you can always do that stuff. Like you can always simulate a whole bunch of activity without learning, then do a bit of learning, and then start again. So there's all that kind of stuff you can do. But the problem of kind of simulating 10 years of learning or whatever is extremely difficult.
Of course. And then the other stuff you were mentioning with I found it extremely flexible that you could like join this with the models of the pyramidal neurons or the other stuff that you mentioned. But you did say that you can't, or maybe I misheard, but compartmentalize that much, or what is the limit there? Like what can GeNN do with?
So there's like there's some tools like Neuron, which people use to sort of simulate very complex models of pyramidal cells. It's kind of the proper geometry of the cells and like sort of 3D diffusion, all this type of stuff. It's kind of a totally different type of simulator in many ways. So GeNN is not trying to be that. But people are sort of developing abstractions of that now, where you have kind of 2 or 3 compartment neurons, and that's the kind of thing Gary and Laura are exploring.
Okay, so that's where GeNN gets to those
yeah, exactly.
extractions.
It's kind of a point neuron simulator rather than a proper multi-compartment simulator.
But the fact that it can be molded into these other variations of stuff, and also the machine learning, the cost algorithm solving that problem that you also mentioned. I thought it was super
awesome. Thank you.
Interesting. So thank you so much. I do have to invite viewers for next week. Not this Wednesday, the next one, April 6th at 17 UTC. Kushal Kolar's talk about Mesmerize, a blueprint for shareable and reproducible analysis of calcium imaging data. So a bit of switch there. Thank you so much, Jamie, for your wonderful talk.
No problem. Thank you for hosting. Thank you for listening, whoever's been listening. Nice to see you all.
Bye-bye.